Thomas Varley, my current postdoc, had a double PhD in computational neuroscience and complex systems at Indiana University, where he was advised by Olaf Sporns. Some of you may know the name Olaf Sporns. He was the founder of what's now known as the field of connectomics. If you understand how things communicate amongst themselves, you may have a unique perspective on how that thing works. And Thomas is a proud inheritor of that tradition, and you're going to hear some of that approach in the Xenobots in a moment. Thomas, who's now working in my lab, has an impressive citation count, number of publications, and a very, very long list of co-authors. So if you have not co-authored a paper with Thomas yet, he'll probably be calling you shortly. And trust me, if he does, you should take that call. All right, Thomas, the floor is all yours.
Yeah, so I'm coming to you guys from a background in neuroscience, and that's going to inform some of the results that you see here today. And we're really introducing this idea of, I have a pointer, xenomics. And this is not a term that I coined. This is something that came out of some of the earlier work on xenobots, but we're trying to establish sort of a rigorous base of study of xenobots. You know, we want the science of xenobots to kind of stand on its own as a field of study in its own right. So that we're kind of calling this xenomics. And I'm going to talk specifically about what we call the informational properties of xenobots. And I'll tell you what that means, but generally what we're interested in is how do xenobots, for want of a better word, compute? How do they respond to their environment, and how do they change their internal functional architecture in response to physical or chemical or other kinds of perturbations? So at the last talk we had before lunch, I think it was Eugene talked about this idea of paradigms. And that's a good segue into what I'm talking about today. Throughout human history, we have defined our sort of technological ages in terms of the materials that we build with, right? Everyone's heard of the Stone Age. We moved into the Bronze Age. Now came the Iron Age. And now we're in what you might call the Silicon Age, right? And all of these ages of technology are built on fundamentally understanding the properties of materials in the world that we can construct with, right? When we made things out of stone, when we understood stone, that's what we could, make tools out of. As we got better at developing metallurgy, we worked on bronze, we worked on iron. Now we have an incredibly high degree of control over microscale silicon transistors, and that becomes the foundational test bed on which all of modern AI runs, okay? This is a fundamental truth that I really want to highlight for you guys, right? That the properties of a material inform what we can build with this. And I'm going to talk you through two very simple, almost trivial examples, but I want to motivate this to then justify all the stuff we do later about xenobots, right? So let's start with wood, okay? Wood is the earliest material that humans probably worked with. There are animals, non-human primates, that still do simple things with wood. And it has properties that make it very useful. It's very low density, you can make a boat out of it, it's very easy to manufacture, it grows in the ground, but it rots, it's it does not survive indefinitely. It's flammable, which makes it maybe not great for cities, and so you can do things like building boats out of it or buildings, but you probably can't build a skyscraper out of wood, okay? Contrast that to another material, ceramics. I'm an amateur potter, I spend a lot of time thinking about ceramics, and it has properties that are very, very different from wood, right? It's non-reactive, unlike wood. It's heat-resistant, unlike wood, but it's brittle. It doesn't bend the way that wood bends. And so you can make crockery out of it. You can use it as thermal insulation on a space shuttle. No one's building a space shuttle out of wood. And you can use it for biomedical implants. But you're probably not going to build a house out of it the way you would with wood. Wood with wood. Which brings us to these guys. And the xenobots are really interesting because they are made of a material that is unlike anything else that humans have worked with throughout our history. Okay? And in fact, some of the work that's been done on it has been done in the absence of really, really deep understanding. Because we can simulate these things, because we can sort of offload some of that cognitive labor to the computer, we can actually start engineering without fully understanding the properties of the thing that we're engineering. And so we're trying to play catch-up with for xenomics. And so like good scientists, the first thing you do is take a closer look at this thing that you're trying to build with. And so can you guys see this? We don't have shades, so some of the pictures are a little bit hard to see. But these are just pictures of the xenobots that were taken by, I think, Pi. These are Pi's stills from some of Pi's videos. And you can see a couple of things right off the bat, right? That, these are composite entities. Every one of these little boundaries is a cell, right? And the cells themselves are composite entities. And so we're already looking at something that is multi-scale, that is fragmented in a way that a blob of metal isn't. And this is, again, very unlike the other materials that we typically work with, right? A xenobot is made up of thousands of individual cells, and not only that, but we saw this morning in Josh's demo, the cells can have different types. You can have stem cells that are undifferentiated. You can have dermal or skin cells. You can have muscle or cardiac cells. And these groups of cells can self-organize into patterns that produce something like behaviors, okay? Metal doesn't do this. Stone doesn't do this. This is a new way of building that leverages the dynamic properties of the material itself. And speaking of dynamic properties, the xenobot tissue itself is also dynamic. It's not just composite, but it evolves through time, right? Cells can talk to each other. They can diffuse signals into their environment and receive them from each other. They can signal each other along what are called tunneling nanotubes, which are much more fast and potentially directed. Cells can change their own internal state, right? They can be alive, they can die, they can become quiescent. And they will respond to changes from their environment by altering their internal states, according to whatever program it is that they are running internally. And so I'm going to show you guys a video, and I have to go behind the desk here for this. Can you guys see this? It's, if not, I'll share it around with you guys. But this is video of a xenobot, and you can see that it's flashing on and off. This is what I mean when I talk about dynamic activity in the cell. Every one of those little lights is calcium, Every one of those little lights is calcium ions fluxing in and out of the cell. And this is going to become, what it is that we build our model of xenobot communication on. Do you guys want to see that one more time, or are we? Yeah, okay, one more time. I can't full screen it for you guys, but.
What's the time scale of this video?
Pi?
Yeah, it's a total of 15 minutes,
Okay.
and each frame captured at 10 seconds.
Okay, and then so, whatever it is, sorry, Google integration slides is not great. Okay. All right, so you guys get to see the URL. I don't know why I did that. But so whatever a xenobot does is going to be an emergent property of the patterns of activity that are instantiated in those cells. Okay? And then so the natural question becomes, what are those patterns? Can we rigorously and mathematically formalize those patterns in ways that will help us better design xenobots and other computational organisms in the future? And so I'm going to play another video for you guys, and I want you to try to see if you can try to pull out a pattern, right? You'll see signals flashing. This is another video from Pi, and see what jumps out at you, okay? This is the video. This is the bot. Now calcium is green, red is actin. And when I watch these videos, it's very clear to me that there is some kind of coordinated activity. You get a little wave, it looks like there's a moment where cells touch other cells and light up, but there's also stuff that looks pretty random. And if you were to ask me to sit down and write this out, I'm not sure I could write out a formal description for you. And so to do that, we are going to turn to techniques originally developed in the field of neuroscience, which is my own home field. I think we're out of video, so I can come back for this. And so in neuroscience, there's a classic suite of analyses called functional connectivity, where you take the brain, you parcellate it up into different regions, you record some kind of temporal signal from those regions, and from these you can construct, a functional connectivity matrix. Okay, every dot here is whether two time series from two different brain regions are correlated. If the dot is red, then they are positively correlated, and if the dot is blue, they are anti-correlated. Okay, this is just a covariance matrix. Then what I've done is I've clustered the covariance matrix so that groups of cells or brain regions that are highly synchronous form their own little communities, and groups of cells that are asynchronous or anti-synchronous are on the off diagonal. Okay? And the first thing that you notice is that this same kind of pattern appears in the xenobots, right? We can take this functional connectivity network and we can cluster it into groups of cells that are internally synchronized and anti-synchronized between them. This was not something we expected. I tried this analysis out kind of on a whim because I had the code, from neuroscience already ready to go. And when this fell out, my jaw kind of hit the floor because what this told us is that the xenobots are not just blobs of cells that have been stuck together. They are actually structured in some kind of non-trivial way. And if you were to do this analysis over and over again, on the top row you've got all xenobots, on the bottom row you'll have all brains for just comparison, you'll see that actually this pattern of highly integrated subsets separated by disintegrated or anti-synchronous bridges is really common in the xenobots in a way that's actually quite similar to what we expect to see in neural tissue. And I don't want to overinterpret these results. I'm not saying that the xenobots are conscious or that they'll do your tax returns or anything like that, although, I don't know, Mike, maybe we can find a way to ask them to do your tax returns, but that these things are complicated. They really are complex systems in their own right. In a way that I think was not obvious to any of us coming into it, given what you saw in the video this morning, where the cells get dissociated and they kind of glom back together. They're somehow able to create or recover, these very, very, non-trivial patterns of organization. So that's pretty cool. We can also look at how those patterns of correlation are embedded in space. And I'm not going to go too far into the details here, but in plot A on the y-axis, what you have is you have the correlation between the strength of an edge and the distance in Euclidean space between those two cells. And what you see is that all of these are negative and all of these are significant, right? This tells us that these patterns are spatially embedded in plausible ways, right? That the closer a cell is to another cell, the more likely they are to be communicating, which makes sense given how we think the biology of these bots works. Likewise, we can see that cells that are within the same community tend to be closer to each other, closer to other cells within the same community than cells that are distant. And the mathematical details of this are not super important, but what does matter is that these are not artifacts. These communities and these structures seem to reflect patterns that we've seen in other biological systems and that are sort of plausible given the hardware that we know the xenobots are instantiating. The xenobots also have higher order information integration. Okay, and this is something that Tristan and I were talking about over lunch, and I don't have time to get into the gory details here, but what this tells us is that if you watch the xenobots through time, right, and for a given value of information and integration, or what you might want to call computation, we actually see very small amounts but significant amounts of what you might call irreducible integration. There's a sense in which the patterns of activity playing out in these xenobots are more informative together than if we were to just parcelate them out into parts, right? The whole xenobot is greater than the sum of its parts in a way that is actually quite similar to what we see again in the neural tissue. Okay? Again, don't want to overinterpret this too much. Again, we're not making any claims about consciousness. Some of you see this little phi symbol and maybe get a little bit nervous. All we're saying is that the xenobots are complex systems that are integrating information in non-trivial ways, which again is not what I think any of us necessarily expected when we started out on this project. I didn't. So, this brings us to an interesting possibility. Can the xenobots act as computers, right? There's a lot of interest in analog computation, and people are working, in spiking neural networks. But xenobots could represent sort of a third paradigm of computation, right? They're going to be analog as opposed to discrete, either silicon transistors or spiking neurons. They are massively parallel, and they're also very, very slow. And so I don't have, a killer app for computation in xenobots, or xenobots as a computational substrate, but, if you have a system that is composite, and its components can be in multiple states, and they change their states in response to perturbations in predictable ways, you're like 9/10 of the way to some kind of computer. You just have to figure out the engineering details. And I will leave it to smarter people than me to actually work those out. Speaking of perturbations, you know, everything I've described right now is xenobots in dishes, you know, just kind of spinning around in circles doing their thing. But I'm going to talk two times about different perturbations that we can do to the xenobots and actually look at how these self-organizing properties change in response to exogenous influence, right? So the first is puncturing. And this is a video that just came out in PLOS Computational Biology with a number of other people on it, particularly Doug Blackiston, Hannah Dromiak, and Caitlin Grasso, who are all the co-first authors. And I'm going to play the video for you. This is a pretty savage video from Doug, where you start with a xenobot, and you can watch, you know, the calcium flashing. It's got little patterns. It's clearly doing something. And at some point here, you're going to see a tremendous flash, which is what happens when Doug stabs it with a needle. And, you know, there's that flash, and you can see that, you know, there's more spiking. There's a little fireworks display that happens and persists even in places where the puncture doesn't happen, okay? So let's go back here. We're pre-puncture, you've got some flashes, there's the puncture, but look down here, right? There is sparking activity happening throughout the whole system that persists throughout, there's one right there, throughout the recording. And if, you know, you kind of aggregate over all the bots, you see, you know, a long transience, the puncture, a huge amount of calcium intensity, and then a post-puncture decrease back to something slightly below baseline. And this is really interesting because, I hate technology. There we go. That puncturing is a response. That calcium flash in response to the puncture is, in animal tissue, a really key part of the healing process, right? If you, you know, stab yourself with a needle, you get a vaccine or whatever, somewhere in your cells there's going to be a flash of calcium, and that is a signal that starts the regenerative process. And so this opens the door to another possibility. Self-healing biobots. If you send a robot out into the field and it breaks, it has a very limited capacity to put itself back together. But if we can leverage, you know, these natural processes that evolution has already kind of built into the tissue, we might be able to create something that is not just robust to the environment, but actually, you know, can take abuse from the environment, heal itself, and continue on. Anybody have any questions so far? I realize I've been going very quickly. Also, Josh, just throw something at me if I'm going too long. Okay. All right, so puncturing, that's one of the perturbations. That's kind of hard to do with MomBot. You know, we don't really have like little needle-wielding drones zipping around MomBot to stab the xenobots. But what we can do, and Sean mentioned this earlier today, is we can use pharmacology to manipulate the structure of the xenobots. And this is very similar to work that's been done all throughout neuroscience, where you have an organism, you give it some drug that changes the properties of its, you know, neural activity, and its behavior changes in some observable way. Xenobots are non-neural tissue, but we can see similar kinds of effects when we give the xenobots the right kinds of exogenous compounds. And so the particular compound that I am most interested in is adenosine triphosphate, ATP. Okay, and everyone knows ATP as the cell, the energetic machinery of the cell. It also has a really, really interesting and much less well understood role as a signaling molecule. And the gist of it is that ATP is not naturally occurring. If you are a cell and you see ATP in your environment, that could only have happened if one of your neighbors started spilling its guts, right? Something happened to your neighbor, and its insides are now on the outside. And so that acts as kind of a warning signal, and it triggers a very, very ancient set of metabolic changes in response to a perceived threat from the environment. We call the cell danger response, okay? And so these are brand new results. This is what I don't want tweeted, because this is like hasn't been peer-reviewed or pre-printed or anything like that. But I have two different measures of what you might call statistical integration of the xenobots, similar to what you saw with the functional connectivity networks. This one actually is just pairwise functional connectivity. This one is more of a hypergraph kind of measure that describes a slightly different flavor of integration. But what you can see is that pre-ATP, you know, there is some baseline level of integration, regardless of how you define it. And then you add ATP, you let them sit for 3 hours, and the total level of integration goes down. You know, it's not a huge effect, but it is statistically significant, and I've put it through its paces, making sure that everything is robust. And what this tells you is that what I think this tells you, I should hedge a little bit here. What I think this tells you is that we are seeing something like the cell danger response in action, okay? That the cells are getting this signal, something in the environment is scary, I am going to enter sort of an every cell for itself kind of mode, stop communicating with my neighbors, and just do my own thing. And that is reflected in the changes in statistical properties of these calcium signaling xenobots. And this is something that if I had millions of dollars in a lab and a plum faculty position, I'd spend the rest of my life thinking about, hint, hint. I think it is quite possible, both for empirical and theoretical reasons, that what we're looking at is something like a dial for multicellular coordination, right? If you think about the evolution of multicellular organisms, we started with free-living cells very, very early on in evolutionary history. And then, we move up this spectrum of emergent coordination to colonial organisms, right? This could be biofilms, this could be sponges, this could be, cells that are working together, but they are not integrated necessarily into a truly emergent whole. And then at the far end, you have sophisticated integration. This is a picture of some neurons that have completely lost all of their individuality and become subsumed into this organ that serves sort of a higher order system, like I think in this case a mouse. And I would like to explore the possibility that ATP, and more specifically, the purinergic system, of which ATP is just one signaling molecule, acts as a dial to drag you up or down this spectrum of emergent coordination. So we've already seen that if you activate purinergic receptors by exposing them to ATP, you kind of get driven down this coordination spectrum, right? The xenobots become less integrated. They become more like individual cells all on their own. What we haven't done, but I would very much like to, is ask, what if we put in anti-purinergic signaling molecules, like ceramide is the most well-known of these? Can we drive the xenobots up the spectrum, increase the level of synchrony or integration in them? I have no idea. That's an experiment that we have to do. But if it is, Mike talks about rebooting multicellularity, this might not just allow us to reboot it, but also control where we are on the multicellular to unicellular spectrum. Big promises, hope we can deliver. And then also, can we abstract this idea and say, okay, well, what else can we look at a spectrum of emergent coordination on? In Josh's lab, we work a lot on swarm robots and stuff like that. And you can imagine a very similar kind of spectrum. On one end, you've got Wall-E, right, who is just happily working, I don't know happily, but he was diligently working by himself on a planet for like thousands of years. On the other end, you have something like a swarm or a flock of drones that make this, make the big constellations that you see, replaced fireworks on the 4th of July. The individuals are simple. What matters is, the picture that they make. In the middle, we could have something, and I couldn't find a robot for this, so I actually just used a picture of a bunch of Marines who are capable of acting as a unit, but also maintain internal autonomy and can make their own decisions while serving the benefits of, the collective platoon that they're part of. And I have some preliminary results that I won't really talk about here using some optimization techniques to show that in silico, we can drive systems up and down this spectrum of emergent coordination. We haven't done anything with robots yet, but that's a direction we'd like to go if we had time and funding. And so, in the last couple of minutes, I just want to talk a little about the future of xenomics and what hopefully, we'll be presenting about at Xenomics, 2 or Xenomics 10 a decade from now. The first is that we have some technical hurdles we have to overcome. Okay. The first is that the data that I work with, this calcium data, is extremely labor intensive. Usually Pi or Doug has to spend hours sitting in front of a microscope collecting this kinds of data. So it's hard to do this at scale. Xenobots want to move. You'll see this in other talks. They're ciliated, which means they want to be dancing around. That makes it very hard to do the kind of image processing stuff that I have to do to reliably segment out cells and get a calcium signal. And then calcium data itself is very low temporal resolution. I come from a background in neuroscience where we're used to getting 10,000 hertz electrophysiological signals, right? Going to something where we have 1 frame every second, that stung. So we desperately need some technological improvements in our ability to image and analyze large number of xenobots in parallel after many interventions. And this is what I'm hoping that MOMBOT will allow us to do. And if we could collect xenobot data the way we currently collect brain data, the sky would be the limit for the kinds of analyses and questions we could ask. I'd like to look at future interventions. So, Mike is famous for all this work on bioelectricity. I'd like to see, what are the statistical properties of these bioelectric interventions. We talked about mechanical stimulation, punctures. Light is something that could be really cool as well. Many cells, have circadian rhythms, respond to changes in the level of light in their environment. And then pharmacological interventions. So I've talked a lot about ATP and sermon. I'm very interested in those because I have another research project looking at some chronic illnesses that are implicated in ATP signaling. But we could look at anesthetics, classic drugs that disrupt emergent coordination. I'd be really curious to see what happens if the xenobots get LSD. Why not? It'd be very cool to see if those same kinds of patterns of disintegration and increased entropy appear in non-neural tissue. And then finally, interactions between xenobots and biofilms, or between xenobots and each other. We're looking at how cells, internally communicate, but none of the math requires that we stay at the cell level. If we had a correct coarse graining, we could look at how sets of xenobots communicate and look at correlations between their patterns of activity. So all of this is, stuff that we could do given, time, money, funding, and would ultimately bring us to my dream, and this could be a career-long project, which is I want a comprehensive understanding of how the biological wetware of xenobots, the cells, the signaling pathways, maybe the genetics, interact to produce the emergent software, right? The behavior, the dynamics, the sensing acting loops that may already exist or we could build into the xenobots from the ground up. Hopefully, I will see you guys all at Xenomics, 10, and I'll have answers to all of these questions. But until then, time will tell. So thank you, huge thank you to Pi and Doug for collecting all of the data for me. It's very labor intensive, and I'm very lazy. I just do math, so they do all that for me. Mike and Josh, who have been incredible mentors and supervisors on all of this. And then I've got to shout out Caitlin Grasso, Hannah Dromiak, and Sarah Walker, who all have contributed to the Xenomics project at various states, even though they're not here in the room with us as well. UVM cuts my checks. All of this is funded by the Cold Regions Research and Engineering Laboratory, and of course, all the members of the Bongard Lab who have listened to me ramble about this so many times now. I don't know how I'm doing on time, but we can do questions.
Got plenty of time for questions.
Okay.
Thank you, Thomas. Questions for Thomas?
Yeah, Hamish.
Great talk, Thomas. Thank you for that. I'm curious, particularly when you mentioned the response of a xenobot to presence of ATP extracellular, and it goes sort of into a survival mode. If I remember correctly, one of the other results from the Levin lab was that one of the first things that a cancer cell does is, from a communications perspective, shuts itself off from its neighbor so that it doesn't get any negative feedback upon growth. Did you see, or do you see any commonality between the ATP-induced sort of survival mode and the cancer cell sort of survival mode cases?
The results you're seeing today came off my computer like the day before yesterday. So there's a lot of questions that we would love to ask that I can't say just yet. I certainly think it's possible. The ATP is the purinergic system is very rich and involved in a lot of different signaling pathways. I could not give you an exhaustive survey of all of them. Maybe Pi, if you want to jump in. But I think it's a very viable hypothesis, and I'd be interested in exploring more. But right now, I can't give you a thumbs up or thumbs down. Yeah.
So you said that there was a principle that you were really interested in at the beginning of your talk, which is had to do with how the materiality or the material substrate of something was a constraint, but also maybe a creative functioning, and like how you can adjust a system. What I'm trying to figure out, like with the work that's being produced by this group, is how the material agnosticism of a system, how materially agnostic can a system be, which is measured, it seems to me, by function, behavior. Like you can rearrange these materials, like different cells, to create xenobots that then perform this behavior that was stimulated virtually before. with regards to the units that you're working with, so the cell is a basic unit of biology, clumps of cells communicate with each other, but other kinds of systems that take a lot of inspiration from xenobots are like silicon, as you were talking about. So what I'm trying to understand is the relationship between the material, like how materially agnostic are systems that you're making? What kind of substrates are you working with? And so it seems to me like that the strong claim about this is that you can be like material substrata. I mean, they matter literally, but they can be very flexible in terms of producing things that produce the behavior that you're asking them to do. But I don't understand how you're working with different units within these flexible systems.
So there's a lot there, and I would be happy to talk with you at length. I will say, just for the purposes of answering this, there's a classic saying in complex systems, all models are wrong, some models are useful, right?
Mhm.
And so I think, in the fundamental level, the materiality matters a lot, right? Biology seems to be arbitrarily complex, and there are going to be times where it's like, okay, I need this particular set of behaviors that only seem to exist in this particular material, right? At the same time, biology also is remarkably good at building coarse-grainable, systems, right? And so there may be times when you say, I really need this particular material property, right? There may be other times where you say, you know what? I don't care about the particular material property. What I care about is, the higher order emergent function. It doesn't matter if it's silicon or xenobots. I don't think that you can say that either approach is universally preferable to the other. And I think a lot will depend on the specific engineering tasks you're trying to solve, right? Sometimes the materiality matters, sometimes it doesn't. The fact that that's a thing in nature is fascinating, and spend a whole career thinking really hard about it. But I don't have a pithy, sort of off the cuff response other than
Thomas, I have a quick question. In the matrix of correlations that you've shown, yes, with the sort of four different or three or four different zones along the diagonal, could they be interpreted, those assemblies of cells or subsets of cells, as some kind of proto-organs or something like that?
That was one of the first thoughts that we had when I first showed these results in our lab meeting. Yes, I think it's very possible that this is some kind of function, this very early functional differentiation starting to happen, and if the xenobots were, if we were looking at the frog embryos themselves, maybe there's a head here and a gut here and a tail here. I don't know. It's difficult because the process of making a xenobot is extremely invasive. They get cut out of their eggs. Sometimes they get lysed into lots of different individual cells and reform. I know pie, sometimes you cut them just out and allow them to do their own thing. I'm not a biologist. I'm a little fuzzy on all the details. So yes, it could be that in the eggs, these turn into organs. In the xenobots, I don't know. They don't live very long. They don't eat or take nutrients from their environment, so they will never develop into, it's not like a Pokemon or something. So I don't know, but it seems like there is a program of some sort that is still running in the xenobot that pushes it towards differentiation. Where it comes from, where it's going, I don't think we know enough to say just yet.
Thomas.
Oh yeah, Mike.
So just to put something on top of that, it actually is possible to feed them, and if you do feed them, the record I believe is 84 days, right? I think that's the oldest we've had. When you push them out that long, it does kind of look like they're doing something, The problem is, the same stuff you use to feed them really feeds a lot of bacteria, and they immediately get contaminated. The 84-day thing was kind of a minor miracle. So we are gonna work on this, but it's entirely possible that once we figure out how to provide nutrients without having them taken over by bacteria, we may actually see that some sort of structure, regionalization takes place, and then this will become extremely important to try to overlay those things. The standard bots, as Thomas said, don't do that, but we do have hope that we can drag them out longer and see what it is that they're turning into, because that's a whole other thing. We know what tadpoles turn into, eventually they turn into frogs. What the heck does xenobots turn into, right? So that's a whole future, a future line of business.
How long do they live when you don't feed them?
When you don't feed them, they live about a week, would you say, 9 days?
12 days,
How many?
10 to 12 days.
And how many cells are in that clump?
Probably about 40,000.
But the parcellation, the computer vision algorithm I use is much, much weaker, much too weak to resolve all the cells. So I'm getting, maybe between a few hundred.
So what are the granulations that we're seeing there? Those are not individual cells? Those are
No,
groups of cells?
they are individual cells. What I do is I take, actually, you're close here. So I take, the video that I showed before. I feed it through, an image registration pipeline, and then I use the cell pose neural network to try to pull out cells. And it'll find, I should have included a picture. It'll find, a patchwork of things that cell pose recognizes as a cell, but a lot of stuff gets left out as well. So we are not getting 40,000 cells. We're getting a few hundred that make it through this image registration and parcellation pipeline.
So to ask a naive question, I mean, you see all this activity in calcium and so on. What is actually moving between the cells? Is it ions, or what's the thing that's causing any kind of communication?
Pi, can I come to you on that?
Yeah, I think exact answer we don't know, but what we are seeing over here is calcium that is coming in and out of the cells. There could be a number of signals that they could be communicating through, like ones that we study generally is bioelectrical signals, so changes in membrane voltages that could be going back and forth. There could be ions that could be going back and forth. There could be chemical signals that could be going back and forth. And also these cells are connected by across relatively far distances through these things called tunneling nanotubes, where they are direct connections but not just adjacent, so far ones.
So when you look at those calcium things lighting up, to answer that question, do you see things light up just next to each other, or do you see one thing lights up and another thing? Yeah, I saw that picture.
Yes.
But that picture doesn't really answer that question. I mean, I'm asking, if you're looking at, if there are multiple mechanisms, one would expect to see some short-range stuff
Yes.
and then some long-range stuff. Is that seeing that? Are those little data points down below, are they something different, or what's the story?
Yeah, so so it's looking at panel A. This is the correlation between, it's a correlation of a correlation, right? So we look at how correlated are the calcium time series.
Yep.
And then we correlate that across against the Euclidean distance between the two cells. And so, these correlations are always negative, right? Which means that in general, cells that are close together in Euclidean space are more likely to be correlated than cells that are very, very far apart. But we do see, statistically significant cell-cell correlations.
I see two little data points there. Are you
each-
thinking those are significant?
Each data point is a xenobot, so this is aggregated over all bots. But within a bot, most of a cell's closest functional neighbors are also its spatial neighbors, but you do find cells that will make long range.
So is there a particular fraction of cells that do that, or?
I would have to get back to you on an exact number, but in non-zero fraction. That's what I'll give you now. Yeah.
And we've also seen waves, waves propagating where you do see it spatially.
So it's like reaction diffusion type waves, or what?
Some of them look like that, not all of them.
There are also duplex waves that happen with some chemical interventions, so there are different kinds of waves that can happen.
So duplex.
Duplex waves. So it's almost like two cells flashing at the same time, but it goes in a wave that spreads across the whole bot. So yeah, there are various different patterns there.
And the types of cells in this, are they all the same in these pictures, or are they multiple types of cells?
We know there are multiple types of cell in there. My statistics don't differentiate between them at the moment. That's sort of on my list of things to try to account for.
They're all subtypes of epithelial cells, so there's several subtypes of epithelial cells.
But didn't you put the cells there? I mean, didn't you sort of filter the cells before you stuck them in this thing?
Yes, and so what happens is we start off with just the epithelial layer, but that layer, the cells are not identical, and they come in, what, 4 or 5 different,
Yeah.
you know,
And you
flavors.
can't mark these cells with one of these cell sorter things and figure out what.
If we wanted them extremely pure, we could, except that I think within a couple days, they would actually try to replenish the diversity.
You mean they would change their cells?
Yeah, they do,
That's
they
right.
do, yeah. So like if you deplete the cells in half, this was an experiment that Doug had done, if you deplete the cells, one type of cell in half, they will repopulate that particular cell in the half that was depleted.
In theory, that could be modulated. If we really wanted to shut that down, we might be able to.
And inside these things, is it, I mean, like, generic cells have some, you know, I don't know, tetratype decahedron, you know, distribution of how many cells there are around a given cell. Is this typical of that, or what, I mean, how many cells are there?
It's packed, it's not hollow.
But like, how
No,
many
no, I
cells,
understand.
how many neighbors does it
Do
typically
you have
have?
14 or something? Is that
I will have
the
to look
typical
back, I
number?
have pictures,
We
yeah.
can reconstruct it. We don't know the
Yeah.
exact number.
Okay.
Yes.
Yeah, I just have a general Xenobot question. You said like around 2,000 cells. Is that like a lower limit for practical purposes, because you can do microsurgery, or like have you made a 10-cell Xenobot, or you know, like how little can it get?
Yeah, Doug has made like small ones made out of 10 or, I think there is a limit, I think 10 or 10, something around 10 to 20 is the smallest limit. Below that, I don't think they aggregate, but yeah, that's what I remember.
I mean, what you'll see, and I'll show some videos of this tomorrow, is that if you make this, there's 2 kinds of Xenobots, one that's made by disaggregated cells and one that's made by just the animal cap. If you make the one out of disaggregated cells, you can see the cells like come together in actually very small numbers, right? And I'll show some of that tomorrow. I don't know that you call it a Xenobot in particular, but they do try to aggregate it to something. The larger scale, you can pile them on as much as you want, but 2 things will happen. First, if you make it too big, it won't swim. So it's too heavy to swim. And if you make it really big, you're going to have issues with oxygen penetration to the middle, so eventually it'll get a necrotic pore and all that kind of stuff. But there is plenty of sort of wiggle room there.
First of all, thank you for this fascinating presentation. Few questions. Have you tried rayoniting?
Sorry?
Rayoniting. Have you tried rayoniting? This will deplete the SR of the plasmic reticulum of calcium. So if you see vanishing all this pattern of calcium-related waste, and if they are important, if they do have a calcium role in creating most of the functional pattern, you should see this, because if you use rayonitin, then you deplete the intracellular calcium store of calcium, and this will affect the behavior of the whole system.
I will
Question.
defer to the biologists on that one, yeah.
We have not done that yet.
Second, you can go the other way around with caffeine.
Yeah.
Just spritz caffeine on the blood and see what happens.
let's do that.
Let's do that. Do these clusters move inside the xenobot based upon the way they spread calcium? I mean, they change intra position. There is a change in positioning of the cells while they do this calcium.
Are you talking about these clusters?
Yeah.
So the clusters are defined sort of at the cell level. So these are pairs of cells that are across the whole recording, highly
Remain
correlated.
stable.
Yes,
Okay,
yes.
and you check also with pH probes, because sometimes when you say that there are clusters of cells that are not synchronized in terms of calcium, the non-synchronized, the one may be synchronized in terms of protein oscillation. There are probes like SNARF, you can test this. This may contribute to a more understanding the connectivity in terms of ion waves inside the
Yeah.
Yeah, absolutely, we have the probes and we are in the process of trying them out.
Yeah, this is, a full characterization of, bioelectric state, pH, and metabolics. This can all be done, and so.
How do you see into the inside of your thing of cells to
You
be
don't.
able to make those pictures?
You don't. I just
So these are only the surface?
these are all the surface cells.
And how big is the thing? You said 40,000 cells, but how big is 40,000?
About 0.5 millimeter.
Okay, so
Yeah.
oxygen, how does oxygen diffuse into the middle?
So these these are loaded with yolk platelets. That's what they use for metabolism most of the time, and then they are sitting in generally the pond water, equivalent of pond water, and so that is sufficient for them to stay alive.
Yeah, I mean, half a millimeter is
Yes.
okay, once you get past the
Yeah.
millimeter is where the problems really start.
But usually it's like 2 microns of actual diffusion of oxygen into the cell, right?
I mean, the standard limit for organoid culture of any kind is you can get to almost a millimeter before you start getting a necrotic limit.
Thomas, quick question, and I'm not actually sure how to frame this, but it seems to me encouraging that your modules have this red-blue differentiation. That seems at this level of resolution as fine as it is, in terms of their ability to maybe continue modularizing in the future if you're able to feed them. But do you have any thoughts on that?
I mean, yeah, I think it's definitely encouraging, and like I said, this was very much a surprise to me to see, like especially like this guy. This is one of the best ones. This really strong differentiation between these two communities. It was not clear to us that this had to be the case. One of the things that I would really like to do, and this would be, this is me sort of asking the biologists for slices of their life they'll never get back. I'd love to record like a xenobot multiple times over the course of its life and see, it'd be very hard to track individual cells, because you get different sides of the bot, we don't have that level of control yet. But it would be interesting to look over the course of, say, 5 days, does this, sort of, red versus blue dichotomy become stronger? Do they modularize over time? I want to sort of make an analogy here. These results, imagine that we've if we're putting this in neuroscience terms, imagine we've dialed back the clock to like 1995, right? Music is different, movies are still good, and this is like the first fMRI image of like a blobby brain that like the amygdala lights up. We've had 30 years of development in fMRI to sort of get neuroscience where it is today. We have not yet had that in xenobots. So a lot of these questions are like, yes, we'd love to do that. We need the level of investment and time and PhD student hours, that neuroscience has enjoyed over the last 3 decades to get to the point where we can really answer them. Yeah.
So, quick question about these images on xenobots. The order, how is the x-axis organized? Is it like spatially adjacent cells? Like how do you order the x-axis?
No, so when I get the raw covariance matrix out, it just looks like a blob of red and white squares. I feed it through something called a multi-resolution consensus clustering algorithm that finds the sorting of rows and columns that makes this pattern most visible. Every row is, like row I represents cell I, and column I is also cell I. These are symmetric across the diagonal, and it's just an ordering of cells that sort of makes this pattern maximally visible.
But I guess my question is, do you see also, if you plot it, are there any correlations between location of the cells and which plot they fall into? Like, do the cells that are close to each other? That's
This is plot B here, and I was unsure how into the gory details I should get, and I can talk to you about.
Those are individual xenobots, not individual cells.
Yeah, so what this tells us is the average delta distance of all the cells that are within a community minus all of the cells, or between a community minus than within a community, right? So in general, you are farther away. The cells that are in different communities are farther away from each other. Yes. And this isn't just this result. We have this in another paper as well. I'm now frantically like, did I write that y-axis right? But yes, in general, we see that the communities are embedded in spatially plausible kinds of ways. Yeah.
Another naive question, if I might. If you just look at random biological tissue and you do the same thing with calcium activation, what does it look like?
We don't know yet, but that's like on my list of dream projects.
That methodology for looking at calcium activation, that's a known biological assay.
Yes, but the parcellating of the set into cells, extracting the time series, doing the functional connectivity analysis, like I would love to do that. And my guess is actually that the xenobots will not turn out to be unique in any particular way. I think these patterns, including like the more abstract stuff, like the integrated information, I think I'm sure this will happen in plants. I'm sure this will happen in fungi. It just happens to be that all of these statistics were developed, not all of them, most of these statistics were developed by neuroscientists, and they've mostly been explored in the brain, which is sort of how I got into this. But my guess is almost anything that you find in the brain, you're going to find at slower timescales in many different kinds of
So
tissues.
the timescale you said here was 10 minutes per frame?
No, it's about 1 second per frame.
Oh, okay, so 10 minutes for the whole
Yeah,
video.
something like that, yeah.
Yeah, it's a 15-minute video and it's 1 frame every 10 seconds.
Okay. Okay. So does the thing physically deform during that time?
Yeah, actually,
It wiggles around.
yeah, there was a video, in the interest of time, I won't pull it up again, but yeah, you kind of watch it kind of go blob a little bit, and that's part of why I have to do a fairly brutal, like, image registration and, motion correction kind of thing.
So it lives for many days. Why aren't you taking video of it, all of its life?
Oh, I wish I could.
What's the problem?
No, because they are incredibly difficult to maintain still because they keep moving. So and for him to do all his analysis, it needs to remain as still as possible.
But it's easy to do image registrations. And what if the thing was moving around? If you're taking a big picture with lots of pixels and the thing's moving around, you just find where it is, right?
So there's two issues. Just playing videos, the problem is that simultaneously getting a giant field of view and having enough resolution is hard, right? So the microscopy is hard because if you leave it alone for a couple of days, it's going to go all over the place, right? So that's one thing.
Why don't you put it in isn't it in some petri dish or something? How does it manage to have room to just move all over the place? Why is it in a petri dish that is in the field of view of the microscope?
Even then, it will keep tumbling around, moving around, even within, so the hole in which I keep it for this is like 1 millimeter or 1 millimeter size hole. Even within that, it will keep tumbling around, moving around.
But so what? I mean, you can, it has, I mean, you can figure out what orientation it's in by just, that's a straightforward machine vision problem to figure out the orientation.
That's great. We are happy to hear that. If that can be resolved, that will be amazing.
Yes,
that has been our bane of existence.
We need to hire some. This has been. Josh has heard me harp about this. We need a really good computer vision person in our labs. My background. I was going to go to medical school. Like I'm kind of trying to wear a lot of hats here.
That problem of what the orientation of a 3D object is, that's a definitely solved problem. In fact, I remember,
Yeah, that's great.
That's awesome.
There is one other thing to think about, which is that if we want to do very long time imaging of the calcium, in order to see the calcium, you're hitting it with excitation, and they get sick from it. They don't like it, and so eventually you're going to kill all the cells. So there's a bit of a trade-off between the more you observe it, the less healthy it gets.
How are you actually observing the calcium?
You hit it with excitation wavelength and you collect the emission, right? So it's a fluorescent report.
Okay.
Yeah, and the excitation light makes the cells pretty unhappy. So the more you image them, the less healthy they get.
That's the reason why the image capture right now is 1 frame every 10 seconds, because the faster we capture, the sooner the cells die. Because you're constantly hitting them with light.